Hello to everyone, and right now I want to check with you a very important theme is a pyrosequencing. Pyrosequencing is actually a special method that's created to understand the whole DNA genome's sequence. Pyrosequencing is such kind of second generation type of the Sanger sequencing, and of course it's more useful. The Sanger sequencing is actually based on the PCR method, and the PCR is actually needs a lot of materials like DNA ATPs or the DNA polymerases, something like this. But pyrosequencing is only needs maybe from the 1 to 15 types of DNA template that of course are the same, and it actually needs not so lot of times because the whole this work is doing only computer, and maybe after one day you're going to have the sequence of a very long DNA template. Here, something like this. Okay, let's go into understand the whole this process. For the first time, you should actually understand that when we have a growing DNA chain, when we add the DNA ATPs, the one molecule of pyrophosphate is actually released. What does it mean? Let's have a closer look on the DNA structure. Here we have our nucleotide is a sugar, is of course a base, and here are the phosphate group. The phosphate group in the normal state has 3 atoms of this phosphate. Like here going to have to be the 3 these phosphate groups too. But when the connection between the phosphate group and sugar is actually created, our 2 molecules is just left this structure, the whole structure, and we're going to have the 2 phosphate groups, like 2 phosphate group that connected with together, and is actually called like the inorganic phosphate groups or inorganic pyrophosphates. Yeah, something like this. And this pyrophosphate is going to create the ATP. ATP is of course created by the adenosine monophosphate plus the pyrophosphate, and it equals 1 molecule of ATP. All of this is created by the enzyme ATP sulfurylase. Then this ATP, you should actually understand that this molecule is of course converted to such kind of energy source, and this energy source are going to help the 2 enzyme luciferase to convert the luciferin to oxyluciferin. What it actually means. Here you can see the molecule of luciferin, the whole structure, and here you can see the oxidized form of the luciferin. And I think that you should clearly understand the oxidized forms and the forms of the reducted, for example, luciferin is this such kind of reduction, and because we have here the hydrogen atoms here and here in the hydrogen bond, and here is the carboxy group. Yes, the hydroxy and carboxy group here, and here we have only the oxygen atoms with a negative charge. Yeah, that actually means that our molecule is oxidized, and during this process the such kind of light is created, and this light is playing a very crucial role in understanding the whole this pyrosequencing method. Okay, then the whole this lights is actually detecting by the charge coupled device is a special program or something like this in our computers, and actually the scientists shouldn't check the whole this process because whole this process is actually detected by our computer. Yeah, and right now I just want to draw you how it works in real life. We're going to have such kind of cell, for example, and this cell is inside of a test tube like here, and this cell is connected with a lot of such kind of tubes. It's so ugly, I'm sorry. And all these tubes actually connected with the tanks with DNTPs, with different DNTPs, right here, here, and here. And here, for example, we're going to have the ATP, a lot of the ATPs, here the TTPs, for example, the CTPs, and here the GTPs, for example. And during one time, such kind of molecules is going inside the cell, and inside the cell we have a DNA template, a single DNA template strand. And for example, the first time, the first is going to be the DATPs. Yeah, then the ATPs just go to the cell, and if this ATPs actually can connect with this single-stranded DNA, this connects, then pyrophosphate is released, and we're going to have such kind of light. Here is actually the net result of this reaction, and like here we're going to have the first, for example, ATP, and when the light is produced, actually we can notice about this in such kind of graph. And for example, the first will be the nucleotide A, the second is going to be C from this tank, okay, C. And for example, if we are going to add such kind of nucleotide that is not going to connect with this single-stranded template, then we have here such kind of enzyme that's called apirase. That is actually is going to denature the such kind of nucleotides that is not connected with a single-stranded DNA template. And then this, for example, DGTP, it just is going to denature, and it will be the structure of this DTP. And the next one is going just to be added. For example, is the DTTP right here. Then, for example, yeah, I'm going to write the whole the sequence right here. And this, for example, this next one will be the ATP, but it's not connected with this one. Oh, I'm just going to write here the sequence too. Then we're going to have the 3 molecules of C, of the DCTP. And here we're going to be the 3 times CCC. The next will be G, then A, then for example T, then C, then T, and again A. And it will be the sequence of this part of DNA. And it's really important to understand this reaction, because in such kind of tasks, tasks, the authors really try to answer you about these methods. That's why it's going to be something like this, and I really hope that you like this video and you actually understand the whole pyrosequencing methods. Thank you for watching, thank you for attention, goodbye.